All right, let's dive into one of medicine's oldest and deepest puzzles, anesthesia. For more than 160 years, it's been the magic that makes modern surgery possible. But here's the crazy part. We still don't really know how it works.
And really think about it for a second. How can this random grab bag of chemicals, things as different as ether, xenon gas, even propofol, all manage to do the exact same thing, safely and temporarily just switch off our consciousness? That's the heart of the mystery we're digging into today.
To really get a grip on this, we've got to go back, way back. See, this isn't just some modern scientific question, it's a full-blown detective story that's been playing out for over a century and a half, complete with red herrings, shocking clues, and some major plot twists.
Okay, so our story kicks off in 1846. Ether works. It's a miracle, but nobody has a clue why. Then, around 1900, we get our first big break, a theory that seems to explain everything, clean and simple. But, like any good mystery, by the 1980s, that theory starts to crumble as new evidence pops up. And that brings us to today, where the whole thing is bigger than ever, forcing us to ask questions about what it even means to be alive.
So that first big break in the case, it happened right around the turn of the 20th century. And it was this incredibly simple, really elegant rule that, for almost 100 years, pretty much everyone thought had solved the whole thing. Case closed. It was called the Meyer-Overton rule. You had two different researchers, working totally separately, who noticed the exact same bizarre thing. How powerful an anesthetic is seems to be directly tied to how well it dissolves in fat. I mean, get this. They were literally using olive oil for their experiments. The connection was uncanny. The more a substance liked fat, the better it was at knocking you out. And this, of course, led to what seemed like the perfect explanation, the lipid theory. The logic was just, it was beautiful in its simplicity. Anesthetics dissolve in fat, right? And our nerve cells are all wrapped up in these fatty membranes. So the drugs must just be sinking into those membranes, messing them up just enough to stop the nerve signals from firing. For a long, long time, it seemed like that was it. But you know how it is in any good mystery, the clue that seems too good to be true usually is. For decades, the lipid theory was the gospel truth. But then, scientists started finding things that just didn't play by the rules. They found these molecules that, on paper, should have been fantastic anesthetics. I mean, they were super fat-soluble. But when they actually gave them to animals, they either did absolutely nothing, or even worse, they caused convulsions. These rule breakers, which they called non-immobilizers, just completely shattered the old theory and blew the whole case wide open again. And this table here, it lays out the problem perfectly. You've got a standard anesthetic, halothane. It's highly fat-soluble, and it works. It immobilizes. Then you have this other compound, a non-immobilizer. It has the same high fat solubility. The old rule says it should work too, but it doesn't at all. This contradiction was the proof. The lipid theory was wrong, or at least it wasn't the whole story. So, with the lipid theory in tatters, investigators needed a new prime suspect. If the anesthetic wasn't just generally messing with the whole fatty membrane, what was its actual target? And that's when attention shifted from the membrane itself to specific things embedded inside it: proteins. This is the huge pivot, moving from the lipid hypothesis to the protein hypothesis. The new idea was that anesthetics weren't just this blunt instrument crudely dissolving into the cell wall. No, they were acting more like a key, fitting into a very specific lock on certain proteins, especially those ion channels that act like the gatekeepers for our neurons' electrical signal. And wow, did the evidence for this new protein theory start to pile up. First, there was this amazing clue from fireflies. Scientists showed an anesthetic could directly switch off the protein that makes them glow. Then, they zeroed in on the ion channels in our own neurons and found that changing just a single amino acid could stop an anesthetic from working. The final proof, the real smoking gun, they actually engineered mice with that one tiny genetic change, and boom, those mice became resistant to anesthesia. The case against proteins was becoming rock solid. But just when it seemed like scientists were about to slap the cuffs on the culprit, these specific proteins, the whole case suddenly exploded in a way nobody saw coming. The mystery, it turned out, wasn't just about people in an operating room anymore. Over a century ago, the father of experimental biology, a guy named Claude Bernard, said this. He had figured out that anesthesia wasn't some unique human thing or even an animal thing. It was something way more fundamental. And he was so right. You can anesthetize almost anything that's alive. Animals? Sure, that makes sense. But also plants. A Venus flytrap will just stop snapping if you expose it to ether. You can anesthetize fungi, algae, even single-celled bacteria. This ability to be switched off seems to be a universal property of life itself.
So why? Why would that be? Well, one fascinating theory is that life evolved with its own internal or endogenous anesthetics. The idea is that organisms might have their own chemicals to put themselves into a dormant state, a way to handle extreme stress, basically a biological off switch for survival. And here is a fact that will just blow your mind. You know, when a plant gets damaged or stressed, it releases a gas called ethylene. It's like a warning signal. Well, get this. That exact same chemical, ethylene, was once used as a safe and effective general anesthetic for humans in surgery. The same molecule. The connection is absolutely staggering. So, after all that, where are we with our 160-year-old case? Have we finally cracked it? Not even close. We've gone from a simple theory about oil to a super specific one about proteins, only to find out this whole phenomenon is woven into the very fabric of all life. The mystery is now deeper and honestly a lot weirder than when we started. One of the biggest remaining clues is just the speed of it all. Anesthesia works in seconds, and you wake up just as fast. That tells us something critical. It can't be a genetic process. That stuff takes way too long. It has to be a direct physical interaction. And here's the final and maybe the most bizarre clue of all. Back in the 1950s, scientists discovered something amazing. If you take tadpoles that are completely knocked out by an anesthetic, just floating there, and you put them under about 50 times normal atmospheric pressure, they wake up. They start swimming around again. You can reverse anesthesia with pure physical force. This points to something fundamental about physics, not just biology. And that's where this leaves us. The search for how anesthesia works has led us from olive oil to fireflies, from stressed out plants to tadpoles under pressure. It's not just a medical question anymore, is it? If the ability to be switched off is a fundamental physical property of all living things, then what does that say about the on state that we call consciousness? The mystery of anesthesia, it turns out, might just be one and the same as the mystery of consciousness itself.